Here is a question that has kept regulatory teams up at night since 2018. How do you prove that a drug isn't quietly carrying a carcinogen at the parts per billion level? You can't test your way to the answer alone. You need a structured risk assessment that follows the molecule from raw material to the finished tablets. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams every day to build and defend nitrosamine risk assessments that regulators actually accept. Today, we are going to walk through exactly how a nitrosamine risk assessment works, step by step, from your starting materials all the way to the drug in the patient's hand. This is one of the chapters that we have covered for our nitrosamine series. So, why does every drug product now need one of these? Rewind to 2018. A routine test on valsartan found NDMA, a probable human carcinogen, sitting in tablets already on the market. Regulators didn't just recall batches, they asked every manufacturer a harder question: have you actually looked? By 2020, the FDA had published a three-step expectation: assess, then test, and then control. Deadlines followed, and when nitrosamine drug substance related impurities or NDSRIs emerged around 2023, the scope widened again. The risk assessment stopped being paperwork and became the entry ticket. So, what is a nitrosamine risk assessment actually? At its core, it's a systematic inventory of every place a nitrosamine could form or enter your product, paired with a judgment about how likely that is. You are not just looking at the active ingredient, you map the synthetic route and its reagents, the solvents, especially recovered ones, the excipients, the water, the packaging, even shared equipment. Think of it as a chemical family tree where every branch gets one question: could a nitrosating agent and a vulnerable amine ever meet here? That question splits into two families, and this is where a lot of early assessments were wrong. The first is the classic small molecule nitrosamines, NDMA, NDEA, etc., that come in form of reagents, contaminated materials, or recovered solvents. The second, newer family is NDSRIs, where the drug molecule's own secondary or tertiary amine reacts to form a nitrosamine unique to that product. Every NDSRI is a nitrosamine, but not every nitrosamine is an NDSRI. And that second family means the risk can live inside the API itself, not just the supply chain. When we score the risk, three drivers do most of the work. First, a vulnerable amine, a secondary or tertiary amine that could be nitrosated. Second, the source of nitrite or another nitrosating agent. And here is the part most teams underestimate: nitrite is a trace contaminant in many common excipients at levels more than enough to matter it. And third, the conditions: acidic, pH, heat during processing, and time on the shelf. When all three factors line up, formation isn't a possibility, it's the chemistry. Not every nitrosamine carries the same weight, so the assessment has to prioritize. Regulators now use a carcinogenic potency approach. The more potent the predicted nitrosamine, the lower its acceptable intake limit, sometimes down to a few nanogram per day. You weigh that limit against the maximum daily dose and how long patients stay on the drug. A once a week short course and a lifelong daily tablet are simply not the same problem, and your control strategy has to reflect that. So we have seen how this plays out. Valsartan and other sartans were the NDMA wake-up call. Ranitidine was pulled from the market worldwide when the molecule proved unstable enough to generate NDMA on its own, and a wave of NDSRI findings has since hit products nobody initially suspected. At Resolve Mass, we have supported sponsors through investigations that started with a single unexpected result and ended in a supplier changes, reformulation, and a refined submissions. The cases look different on the surface. The pattern underneath is almost identical. Someone assumed instead of assessing. So here is how whole thing runs end to end. Step 1, scope and inventory, map every material and process input. Step 2, the risk assessment itself, decide with justification where formulation is credible. Step 3, confirmatory testing, where risk is real, develop and validate a sensitive method, usually LC-MS, MS, GC-MS, and actually measure it. And step 4, set the acceptable intake limit and assess the toxicology. Step 5, control and report CAPA, supplier controls, specification changes, and the documented story the regulator reads. Something most explainers skip. Step 1 and 2 decide everything downstream. Scope it wrong and you test the wrong things. So what should you walk away with today's discussion? Risk assessment traces the whole product, not just the API. Nitrosamines arrive from the supply chain and can form inside the molecule itself. Potency and dose set the limit, and the assessment, not the assay, is where the real work happens. If you're working on an IND or ANDA or a marketed product and you need a CRO partner who treats nitrosamine risk assessment as the real science rather than a tick box exercise, Resolve Mass Laboratory supports small and mid-sized pharma companies from first inventory all the way through confirmatory testing and submission. Subscribe this video channel so that you don't miss all other informative videos, and it helps us to create more and more videos. Drop a comment on the topic that you want us to cover on the video. Thank you so much for watching this session. Goodbye for now and see you in the next one.